Hi, my name is Nowrosh and I am a PhD candidate at Bowl Lab from Department of Biology. Today, I am going to talk about my current research project entitled as Peptidoglycan Recycling Promotes Outer Membrane Integrity and Carbapenem Tolerance in Acinetobacter baumannii. So as we know, the antibiotic treatment failure is one of the tragic reality experienced throughout the world. Besides resistance, there are another potential phenomena of the treatment failure, and that is called antibiotic tolerance. The antibiotic tolerance is basically defined as the ability of the bacterial population to stay survive for a long period of time in the presence of the bactericidal drug. And this tolerance is considered as the stepping stone in acquisition of true resistance. So, some of the previous studies showed that the gram-negative clinical isolates of Enterobacter C show high level of beta-lactam tolerance, and they form a unique cell wall lace-like structure that is called the spiroplast. However, the molecular factors that promote the spiroplast formation is not well understood. So here, our goal is to define the tolerance factor to understand how bacteria survives for a long time in the absence of the cell wall. So to understand this, we used Acinetobacter baumannii. Acinetobacter baumannii is an opportunistic pathogen and rapidly acquires resistance to all the conventional antibiotic. So we reason that the factors promoting carbapenem tolerance may be widespread among susceptible Acinetobacter baumannii strains. So to see susceptible lab strains of A. baumannii show meropenem tolerance like Enterobacter C, we determine the colony forming unit per ml. In the figure 1a, as you can see, all the susceptible lab strains that actually can survive and shows high meropenem tolerance. And in figure 1b, we have done the microscopic analysis of this tolerance cell, and we found that they form spiroplast after 12 hours in the presence of the meropenem. Next, we have done RNA-seq to define how transcriptional as alternation associated with the spiroplast mediated tolerance. And figure 2 represents our RNA-seq results. We found that the meropenem tolerance is associated with the downregulation of the porins and the activation of the efflux pump, PG recycling genes, and also the outer membrane lipoproteins. However, one of the limitations of the RNA-seq is differentially regulated gene does not encode in bacterial fitness. So to pinpoint the bacterial fitness during meropenem tolerance, we have also done TN-seq screening, and we validated our TN-seq results. And you can see in figure 3a several novel genes associated with the outer membrane integrity maintenance genes such as the OMP, LPXM, and the periplasmic and cytoplasmic cell wall acting gene PBPG and LDTK are involved in meropenem tolerance. And when we knock out these genes, we found the mutants shows high susceptibility than the wild type and the complementary strain and their untreated controls. And in the figure 3b, we found that we showed the ethidium bromide permeability assay, and we found that all the mutants accumulate high ethidium bromide permeability than the wild type and the complementary strains, suggesting these genes work as the cell envelope barrier function and limit the meropenem entry. However, we were not sure what is the genetic link of the PBPG and LDTK in carbapenem tolerance. So next, to explore which specific pathway is involved in meropenem tolerance, we define the biochemical activity of these genes. And we have found, and based on the figure 4a, based on neuropeptide analysis, we have found that the PBPG is active against the pentapeptide, and it has both DD-carboxypeptidase and endo-carboxypeptidase activity in both log and stationary phase. Whereas in figure 4b, we have found that the LDTK is a LD-carboxypeptidase, and that is located in the cytoplasm and active against the tetrapeptide, and it releases the tripeptides in the environment. And the PBPG provides the substrate for this LDTK. So this suggesting that this PBPG and LDTK, they are working together and actively involved in PG recycling. So in conclusion, together our study suggests that both outer membrane protein and the PG recyclings are tightly linked together and involved in meropenem tolerance. So that's all for today. At last, I would like to thank all.